Now, what if the invaders actually get past the barriers and past the internal defenses, and there's just too many invaders, too many pathogens for the internal defenses, the non-specific innate defenses to deal with? Vertebrates have an advantage where we have what are called adaptive immunity. And that's where our immune system can respond to one specific type of invader at a time. Now, for adaptive immunity, well, we're going to divide that up into two types called humoral immunity and cell-mediated immunity. And what that really means, humoral immunity is just the immune response that happens in our body fluids, in our blood and lymph. And cell-mediated immunity happens within the cells and tissue. And it's just kind of traditionally been divided up that way. We're going to start out with imagining a pathogen invaded. Let's say it's a bacteria. So here's a bacteria cell in green, and it has on its surface certain protein markers that identify it as an invader. These markers that our immune system recognizes as invaders, we're going to call those antigens. Okay, so let's imagine this pathogen, this bacteria, got engulfed by a phagocyte or a macrophage. There's several different kinds of cells that will engulf invaders. So let's say a macrophage is engulfed this bacteria. The next thing that's going to happen is it's going to digest this bacteria. So lysosomes within that macrophage that are filled with digestive enzymes are going to merge onto that bacteria cell membrane and dump out the enzymes and dissolve that bacteria. So there they go, the enzymes are dissolving the bacteria, and finally what's left at the end is a vacuole or a vesicle that contains the remains of that bacteria, including the antigens. Now those antigens are going to be taken and exported to the surface of that macrophage cell. And then we're going to call it an antigen presenting cell. So here's our antigen presenting cell with the antigens displayed on the surface. Now the key for active immunity is for our cells to recognize what's an invader and what's not an invader, what are our own cells. So to do that, we're going to combine these antigens from the pathogen with marker proteins from our own cells, from this antigen presenting cell. I'll show our own marker proteins in purple. So our own proteins, we're going to call a group of proteins that identify us, our tissues versus invaders, is called the Major Histocompatibility Complex proteins. Huge name, MHC, Major Histocompatibility Complex. Okay, as long as one of our immune system cells sees that protein, it's going to say, oh, that's us, that's one of our cells, don't kill it, that's good, that's one of us. But if it sees that in combination with another invader protein, it'll say, okay, that's the kind of cell that the invaders are using. That's what I've got to destroy. So one of the most critical types of cells in our immune system, our immune system is divided up into leukocytes, or white blood cells, and lymphocytes. And there's several different kinds of leukocytes and lymphocytes. The two most important lymphocytes are called B cells and T cells. One of the most critical are called helper T cells, and what they do is recognize the antigens that are presented by these antigen presenting cells and then start advertising for the rest of the immune system cells to start attacking that particular invader. So here's a helper T cell, and you can see it has on its surface of its cell receptors that match the shapes of the major histocompatibility protein and the antigen from the antigen presenting cell. And as long as it matches in combination, it'll recognize, okay, this is one of myself presenting what I need to attack. And that's how essentially it's how our immune system can recognize specific foreign invaders. Now when the helper T cell binds on to those MHC and antigen proteins, that's going to stimulate the helper T cell to start dividing by mitosis into a group of helper T cells, and it's also going to start releasing chemicals to attract other immune system cells. As these chemicals, called cytokines, diffuse into the surrounding area, diffuse into the bloodstream and travel around the body, they're going to start causing new kinds of cells to become active. And there are different kinds of cytokines. For example, let's say this first kind, this brown cytokine chemical, it's going to activate B cells. And let's take a minute now and explain the other major types of lymphocytes, the B cells. B cells recognize antigens, and they don't have to be antigens that are presented to them by antigen-presenting cells. So let's say floating around in the bloodstream, there's some of these antigens from a destroyed bacteria that's been invading, say. They're floating around, and a B cell would recognize those. B cells have on the surfaces of their cells receptor proteins that match the shapes of these antigens. So this particular B cell has the right shape, the triangle-shaped receptor. Now, there could be another B cell floating around somewhere that has a different shape of receptor, little goalpost receptors. And another B cell somewhere that has little rounded receptors. The thing is, there's hundreds of thousands of possibilities of different shapes of receptors. Let me explain how these receptors are built, where they come from. Because each B cell has its own specific shape of receptor, so it'll only recognize one specific type of antigen. Let's say in the bone marrow, where blood is built to begin with, we have a pre-B cell. It's not a B cell yet. Here's the nucleus. And of course it's going to have some DNA inside the nucleus. But we need several pieces of this protein. We're going to assemble it piecemeal, one section at a time. Let's say this first piece of DNA builds a section of a protein that looks like that, has that shape. The second section can build one of the arms, and the third section build another arm. But however, let's imagine we have lots of different sections of DNA inside this nucleus, and each one can build different shapes. Okay? Say the top one builds this shape, the next one 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 that shape. So what we do is select each B cell before while it's still a pre-B cell, will turn off certain of those genes, or certain segments of that gene. So say, for example, we eliminate that one, and the bottom one, and this one, and what's left is the section that makes the stem of the receptor, and then each arm. So we would have a stem that looks like this, the top section would look like this, and the bottom section like that. And we would get a particular kind of receptor on the surface. If we had selected a different gene, say this one, if that gene segment was on and this one was turned off, we'd build a different receptor. It would look like this. So that's how we get B cells with all the different kinds of possible receptors, and it's just simply a way of mixing and matching, sort of like playing Legos with within the DNA to select which kind of receptor you want to build. Once the B cell is matured and released from the bone marrow and circulating in the bloodstream, if that B cell happens to connect to an antigen, it will stimulate that B cell to start dividing by mitosis and making a whole bunch of new cells. Each of those new cells is going to have the exact same kind of receptor that particular B cell makes. So here's a bunch of B cells, and their job, we're going to call these, as they specialize, we're going to call these plasma cells now. And instead of retaining the receptor sites on the cell surface, they secrete these proteins out into the bloodstream. They just build thousands of them every minute and then dump them out into the bloodstream. So instead of the receptors staying on the cell, they dump them out. Here they are, dumped out into the fluid. They go floating around. And of course you know what we call these kinds of proteins. That's right, they're antibodies. Now antibodies protect the body from invaders, from this particular kind of invader that was carrying this specific antigen, by attaching onto those invaders and preventing them from entering cells. Since the antigens are globbed up with antibodies, as soon as the antibodies attach to the antigen, it makes them more an easily recognized target by other immune system cells. It's almost as if the antibodies are attaching on and waving, hey, over here, here's an invader. So these immune system cells rush over and take care of the problem. Okay, so antibodies are a very critical part of our immune system, of our specific active immunity. Also, in addition to making these plasma cells, these B cells will divide, and some of these B cells will not be used during that particular battle, but they'll hang around, wait around, in case they're needed later on. If this same invader shows up again, they'll be ready. So these cells can circulate around in the bloodstream for years. We're going to call these memory B cells. And they'll produce that same exact kind of antibody in case it's ever seen again, if that invader ever comes into the body again. As the helper T cell created more helper T cells, they secrete more cytokines, which attract more B cells to the area, so the chances of finding the particular B cell that has the receptor that matches with the specific antigen that it needs is increased, and our immune reaction can be much faster. Okay. Also, if you'll notice, these helper T cells do the exact same thing as the B cells in producing some cells that will stick around later and wait for years in case that particular invader ever invades the body again. And so we'll also call those memory T cells, or memory helper T cells. Let's imagine a different kind of cytokine chemical released by the helper T cell also. So here's another cytokine released by the helper T cell. It's going to activate what are called cytotoxic T cells. Cytotoxic T cells have to also be presented the antigen by that antigen presenting cell. So here's a cytotoxic T cell being presented that antigen along with the major histocompatibility complex protein from an antigen presenting cell. Okay. Then when the cytokines from the helper T cell reach that cytotoxic T cell, it will activate it to divide and make more cytotoxic T cells, some memory cytotoxic T cells. And what these cytotoxic T cells do is release chemicals that will kill the invading cells. Cytotoxic T cells release at least two kinds of chemicals that will destroy cells. One will diffuse into a cell and cause it to initiate apoptosis, programmed cell death. And they'll also release chemicals called perforins that will assemble together and produce holes within the cell membrane of the other cell, so that fluids start to rush into the cell and destroy it by osmosis. This can be a real advantage because, again, some of these antigen presenting cells, they engulf an invader, they engulf a pathogen, maybe haven't been able to kill the pathogen yet, so the pathogen is still inside the cell. But as long as they can approach a cytotoxic T cell and basically say to it, hey, I've got an invader inside me, kill me, you know, that helps our immune system to destroy the invaders that are inside cells and trying to hide out there. So cytotoxic T cells are also an important component of our immune system.